Okay, so our next speaker will be Ying Jiang, and he will give a talk entitled Changed Epigenetics Caused by Neonatal Fasting Stress Protect Chickens from Future Fasting Stress.
Thank you very much for the introduction. And my topic today will be the epigenetic change caused by neonatal fasting protects the chicks from future fasting stress. So, introduction. Unfavorable nutrition conditions such as fasting during the neonatal period can very easily upset the set point of the neurochemical set point. So this can cause not only an acute metabolic syndrome, but also some severe disease in the later life. So in our lab, our curiosity is how does the early fasting affect has a long-term effect in the body, such causing severe disease in the later life. Some studies so far has suggested that stressor will cause epigenetic changes on the chromosome, which will affect the gene's transcription. So one of these epigenetic marks has been identified on the histone 3 and terminal tail, and the lysine 27 is the key site which has been tightly related to the transcription inhibition. So this is a graph comparison between the euchromatin and heterochromatin. The euchromatin is a structure of the chromatin which does not have the methylation on histone 3 lysine 27. So in this case, the chromatin is kind of like in a loose structure, so the polymerase can very easily get onto it and start transcription. However, for the heterochromatin, the methylation has been put onto the histone 3 lysine 27, so the histone will make the chromatin become compact, so the polymerase cannot get onto it, so the transcription will be totally shut down. And the PRC2 complex is a very specific histone 3 lysine 27 methyltransferase. And three key components has been identified in its active site, which are the EED, SUZ, and EZH2. And the EZH2 is the only protein in this PRC2 complex which has the methyltransferase activity, while the other two serve as an anchor protein which bind to this PRC complex onto the chromosomes, so let the EZH2 form its methyltransferase activity. So in the previous study, our lab has shown that after 24-hour fasting, the three-day-old chicks will instantly show an methylation status on the histone 3 lysine 27 in the hypothalamic nucleus called the PVN, which is an energy center regulates the energy homeostasis in the individual body. So our question now becomes, how long will these epigenetics changes exist on the chromosome, and what's the function of these epigenetic changes on the chromosome? So to answer the first question, this is the experimental design, and for the animal we have first a control group which has 18 chicks inside. They are all male broiler, fed at liver temp, never been fasted. So this is day 4, day 11, day 41. So at each time point, 6 chicks will be sacrificed and the PVN will be collected. And for the fasted group, also has 18 chicks, but they will be fasted on day 3. The black diamond indicate the fasting for 24 hours. And 6 chicks will be sacrificed at each time point, day 4, day 11, and day 14. And the PVN will be collected too. So we are going to use the Western blot testing the total histone 3 protein level and the methyl modification on histone 3 lysine 27. And also using the qPCR testing the mRNA level of the EED, SUZ, and EZH2. All of them are the components of the PRC2 complex. And the statistics will be performed in the jump and using the simple t-test, trying to compare the difference between the day 3 fasting and the control group. So here's the data, and the blank column indicates the control, while the blank column indicates the day 3 fasting. This is the total histone 3 protein level, and you can see right after the fasting, a significant boost of the histone 3 level happens inside the body, and also the methylation status, these two. This is dimethyl, this is trimethyl, and significant increase the level happens in both of these two kind of form of modification. And also this modification lasts until day 41. You can see a significant decreased level of dimethylated H3K27. And also for the qPCR, we tested the PRC complex, and for the EED SUZ, you can see a significant increased level right after 3-day fasting. And also for this too. And also on day 41, which is a pretty long day after day 3 fasting, effects still remain on day 41 for this EED significant increased level, and for this EDH2 significant increased level. So this result indicates the day 3 fasting can induce a very rapid epigenetic changes, resulting in a methylation of histone 3 lysine 27 and the key components of the PRC2 complex in the neurons in the PVN. And also, in addition to these rapid changes, the following change can also happen. For example, day 11, which is like 7 days after the fasting, these two parameters remain decreased. And for this day, on the day 41, which is like 38 days after the fasting, the 3 parameters remain either decreased or increased. So the conclusion is the epigenetic changes can occur right after early fasting, 24-hour fasting, and these changes may result in alternation of the neuropeptide level later in life. And trying to confirm this statement, we use a very specific anorexigenic factor, which is the BDNF, which is short for the brain-derived neurotrophic factor, which also has an effect in the neural survival and synapse formation, this kind of like brain maturation requirements. So this is the protein level of this BDNF, mRNA level of this BDNF. You can see right after fasting, a significant decrease in both protein and mRNA level. And on day 41, which is 38 days later, after this 3-day fasting, a significant decreased level of mRNA still remain there. So this part of the result agree with the previous result. And the BDNF's protein and mRNA were rapidly affected by day 3 fasting. And on day 41, the BDNF's mRNA remains significantly lower compared to the control. And this indicates that the early fasting can have a long-term effect inside the body. And the second question is, what's the function of these inhibitory marks on the chromosome? So why the body want to put all these onto the chromosome? So this is the experimental design trying to answer this question. And for the control, we have 6 chicks, and all of them have never been fasted and sacrificed on day 11. And for the second group, single fasting on day 10 for 24 hours, the black diamond, and sacrifice on day 11. And for the third group, they will be initially fasted on day 3 for 24 hours, and again fasted again on day 10 for 24 hours, and sacrifice on day 11. And PVN will be collected from all these chicks on day 11, and same method, Western blot qPCR, testing the same parameters, and the statistics will be performed in the jump and using a one-way ANOVA 2P test. So here's the result. The blank column indicates the control, the green column indicates the single day 10 fasting. You can see right after the fasting a significant decrease in total histone 3 protein and methylation modification on histone 3K27. These two. However, when you compare this double fasting to the control in the single day fasting, you can see the histone 3 protein level has been brought back to the control level, and same for the methylation modification on histone 3K27, but not on this one. For the qPCR, EED SUZ showed same trend as the Western blot data. You can see right after fasting, significant boost of the EED and SUZ. However, when you give day 3 fasting before a day 10 fasting, the EED level will be bring back to the control, and also the SUZ, the level will be bring back to the control. So this part of the result indicates 30-day fasting may ameliorate the effect of later fasting. And this suggests that epigenetic changes caused by early fasting may induce a protective mechanism in the body, which maintain an internal homeostasis after the suffering with similar stress. So trying to confirm this idea, we still pick the BDNF gene. This is the control, and this gray column is the day 10 fasting only. You can see significantly decreased in both protein and mRNA level. However, when you give a double fasting to the chicks, you can see the protein and the mRNA level is ameliorated and bring back to the control. So this part of the result indicates the day 3 fasting was able to attenuate the changes in the BDNF's protein and mRNA caused by fasting later in life. And the early fasting may induce a protection from a similar stress later in life. So in this study, both day 3 and day 10 fasting can induce a very rapid epigenetic changes in the neurons. And the epigenetic changes caused by the early fasting may induce a protective mechanism for the neurons in the BDN. One of the early studies using a different animal source, C. algan, reported that they behave differently to the cultivation temperature after initial starvation experience. So combined with this, with our result, this suggests that the protective mechanism I showed up here may be a stressor specific. So for the future study, because the C. algan experiment using a different animal source, which might be animal category specific. So we have no idea whether this idea is true or not, whether the chick will respond with. So we would like to ask, does early stress only protect the body from the same stressor, or it has a cross effect and other things like, for example, what's the changes of the other feeding regulation factors? So here I would like to thank my committee member, my lab, the department, and Dr. Gilbert. So any questions?
I have a question, ma'am. You showed global changes in epigenetic modifying enzymes and an abundance of histones. Do you have any evidence to show that there are changes at specific gene loci, for example, your BDNF gene?
No, this is the only protein. This is the only gene site I tested. That's why, like in my future study, I would like to test other feeding regulatory factors. How's the epigenetic changes on their genes?
But those changes were global, not specifically on your BDNF gene or on its liver, for example.
What do you mean by global?
Global, I mean like different gene may have different dynamic changes on its histone distribution. So for the global, this only indicate like this is the train of the cell inside the cell. But for different factors, like for example, I showed up in anorexigenic factor, but I have no idea how about feeding promoting factors means. Maybe they've been promoted, and for the anorexigenic factor, they've been inhibited. So the dynamic of the histone protein distribution on these two different kind of genes will be different. But overall, the trend will be there. This is the global means.
So by global, you didn't really mean the epigenome itself. You're talking about specific response.
Not specific.
Yeah, so I'm confused. I mean, you have the epigenome, right?
Yeah.
And so you have multiple factors there. But you're really looking at this BDNF,
Yes.
which is specific.
Yes.
So I'm not sure how where you're going with the global term.
We're trying to see like whether this specific thing has a very specific connection to the global one, or whether this is like tightly connected, show a similar trend, or it's just
Okay.
Any other questions for our speaker? Let's thank our speaker.